We're going to start out looking at the Calvin cycle, which is basically the fixation of carbon. This is the way that carbon dioxide is picked up in the biological world and stored. So when we're looking at the Calvin cycle, we're actually looking at the process of picking up the carbon dioxide. It starts out with a chemical called ribulose 1,5-bisphosphate. We call this RUBP. You can see it's got 5 carbons in it. It's got 2 phosphorus groups on the 5 carbons. There is an enzyme called ribulose 1,5-bisphosphate carboxylase, which was known as Rubisco, which is the most prevalent enzyme on planet Earth. And it takes this process where the ribulose 1,5-bisphosphate is picked up. To it, it adds water and it adds carbon dioxide. The first stable intermediate you get out of that is called phosphoglyceric acid. It is a 3-carbon unit. So basically, out of one ribulose 1,5-bisphosphate and 1 carbon, you get 2 of these. Now, this is changed using a phosphoglycerate kinase and ATP. So you can see we're pulling in ATP out of this to make a 1,3-bisphosphoglyceric acid. This is also, both of these chemicals are also part of glycolysis. So this makes material that is used in glycolysis as well. You then use NADPH and a phosphorus coming out of it with an enzyme called glyceraldehyde 3-phosphate dehydrogenase, and you get glyceraldehyde 3-phosphate. This gets modified using a triose phosphate isomerase to give you dihydroxyacetone phosphate. Now, all 4 of these, again, are in the part on glycolysis. From the dihydroxyacetone phosphate, there is an aldolase that combines these 2 chemicals, the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, to give us fructose 1,6-bisphosphate. Now, fructose 1,6-bisphosphate, it's also the precursor for fructose. So we now have chemicals that can be used within the plant. There's a phosphatase that takes a phosphorus off of this to give us fructose 6-phosphate, and this can combine with the glyceraldehyde 3-phosphate using a transketolase and give us a xylulose 5-phosphate and an erythrose 4-phosphate. Now notice, out of these, we're taking a 6-carbon and a 3-carbon to get a 5-carbon and a 4-carbon. All of these serve as precursors for other pathways as well. The erythrose can combine with the dihydroxyacetone phosphate and an aldolase to give us cetylheptalose 1,7-bisphosphate, again a 7-carbon carbohydrate, with a phosphatase will give rise to cetylheptalose 7-phosphate. The cetylheptalose 7-phosphate combines with the glyceraldehyde 3-phosphate, so now we've got a 7 and a 3, it's going to give us a 10. Then out of this we get a ribose 5-phosphate and a xylulose 5-phosphate. The plant is manipulating these chemicals to get precursor molecules for other things. The xylulose 5-phosphate gets changed by a phosphopentose epimerase, and ribose 5-phosphate gets changed by a ribose 5-phosphate isomerase, and that will give you ribulose 5-phosphate. This then gets phosphorylated using ATP to get us back in the beginning where we've got our ribulose 1,5-bisphosphate. The ribose phosphates, being 5 carbons, can be used for the formation of RNA or other materials like that. So we do have precursor molecules in here being created all the time. Out of this, you can see it's a rather complex pathway system where one gives rise to another thing, and then we can add things together and we can break them down. But that is the essence of the Calvin cycle, which is really the principal way that carbon is picked up in the environment and converted into organic carbon forms.